Thanks for joining us today. My name is Loren Wold. I'm the editor-in-chief of the FASEB Journal and FASEB BioAdvances, and it gives me great pleasure to introduce the new deputy editor for FASEB BioAdvances from our competitor school at that state up north, Dr. Thomas Sanderson. So thanks for joining us today. And today we're just going to spend a little bit of time of getting to know you and your background in editing and in being involved in journals, as well as your scientific background. So why don't you start by just giving us a little background of yourself and your lab and what you work on?
Sure. Yeah, so for those of you that don't know, that school up north is also called the University of Michigan, and that is where I have my research lab. So my lab is focused on understanding how acute brain injury progresses after stroke, after trauma, after a lot of different types of severe critical care injuries to the brain. We really focus on the molecular mechanisms involved in the progression of brain damage, and then we take our understanding of those mechanisms and we try to design new therapeutic approaches. And we do a lot of testing of new therapeutic approaches to try to limit brain damage and eventually get something to clinical use that can help protect people that are suffering from either a stroke or a traumatic brain injury. So that's my research. My editorial background, I started at Life Sciences as an associate editor about 5 years ago. And we worked to bring that journal to a new level of rigor and reputation. And then we moved here to FASEB BioAdvances and FASEB Journal, where I'm an associate editor at FASEB Journal. And then I've just recently been added as the deputy editor of FASEB BioAdvances. Really, the goal here is to try to publish the best, most rigorous research that we can get our hands on, and really be a great place for the research world to publish their best work.
Great. So as you mentioned, you were brought on to the journals, specifically FASEB BioAdvances, to help really elevate the journal. I think the journal in the past had there wasn't much of an identity that it had, and so I think you would agree our goal has been to help define that identity. So what would you think if you had to explain, what is a typical journal article in FASEB BioAdvances? How would you explain that?
Yeah, so FASEB BioAdvances does just like just like FASEB Journal. It has a very broad readership. It has a very wide area of interest. Now we really focus on science that is quality science, but a lot of these research papers that we're getting aren't necessarily either as large in scope or as perhaps high in significance, or at least the significance isn't as clear to date, as you would get in a FASEB Journal article. So FASEB BioAdvances is really focused on capturing good research, solid, quality, rigorous research that may not be a thematic fit or a mechanistic fit for, say, FASEB Journal, but it deserves to be in the scientific record. It's something that is valuable and should be published. And we try to really capture all of those types of research research manuscripts and publish those in BioAdvances.
Right. So what some people may not know is that if an article is submitted to FASEB Journal, we may recommend that it actually be transferred to FASEB BioAdvances. Obviously, anyone can submit their articles to either FASEB Journal or FASEB BioAdvances. We call that sort of de novo, or new submission to the journals. What happens, maybe you can describe to the listeners, if I transfer an article to you and say, it's not quite mechanistic enough for FASEB Journal, what happens on your end?
Yeah, there's a couple different ways that these papers are going to reach BioAdvances. Obviously, de novo submissions. is just somebody that directly submits to BioAdvances. But the majority of the manuscripts we get are transferred to BioAdvances from FASEB Journal at some level of, after some level of either editorial or reviewer review. Okay, so a lot of times there will be a manuscript that comes into the associate editors at FASEB Journal, and that associate editor may immediately realize that this is not a great fit for the theme of FASEB Journal, and then will, but it's quality research, and they think it'll be a good fit for FASEB BioAdvances, and they'll directly send that to BioAdvances for consideration. Those papers will come to us. We will send them out to review. We'll get reviewer critiques back, and then go through the normal publication process that you would see at FASEB Journal. We also sometimes get papers that went out for review at FASEB Journal, and the reviewers come back and say, well, this is nice science. They do really clean experiments, but it's not really the scope or size that we would expect to publish in FASEB Journal, and might not be a good fit for the journal. Those will go to FASEB BioAdvances, and we can at that point take that manuscript and say, okay, if you do a response to reviewers and adequately address the concerns of the reviews, we can consider this for publication at FASEB BioAdvances. So there's a lot of different ways these manuscripts can get there, but the end goal is to try to find quality science that isn't quite the right fit for FASEB Journal, and give it a home in BioAdvances.
Great. So you've touched on this in some of your comments, but in your opinion, what makes a good publishable article?
Really, it comes down to the data. So, the first thing that we look at when these manuscripts come in is the figures, the data that came with the manuscript. And is it of quality science? Is it fully powered study? Is it rigorous? Does it have all the proper controls? Is it presented in a manner that's readable and quality, and will contribute to any field, be it even a small field that may have only be of interest to a small subset of scientists, but it's still of interest and of importance. So it really does just come down to that quality, to that rigor, much less so than the potential impact or significance of the study. It's much more a quality assessment for us at BioAdvances.
Great. So last question I'll ask is, a lot of times when I meet authors, I'll say to them that my commitment as an editor is to provide a fast, thoughtful, and rigorous critique of your science. Obviously not personally, but through the editorial process. So what is your commitment to authors that are submitting to FASEB BioAdvances?
Yeah, really the goal is to get a fair and productive review, to find out if this manuscript is a good fit for BioAdvances, and if it's not right away, what can we do? What types of experiments or changes could be made to make this publishable in BioAdvances? We really want to help the authors get to the point where their study is ready for publication. And we'll be very blunt to start if it's just not going to be a good fit from the beginning. But if we bring it in for review, we want to try to get it over the finish line. And we do that by trying to find the best reviewers that are committed to investigations in similar topics. And then I personally will go through the reviews and make sure that they're thoughtful and useful, and help that author try to get their manuscript to the point where we can publish it in FB. So we like to be a home for studies that maybe aren't large in scope, right? So sometimes you might have an experiment or a series of experiments that are they find something very interesting, but it's not necessarily the whole story, right? It's a piece of the puzzle. It doesn't mean it's not publishable or it's not something that should be in the scientific literature. And so we try to get those types of studies in publishable form and try to get those into FB. Yeah, that's really it. I just try to do my best to shepherd these through the editorial process and get those studies to a point where they can be published with us, or if not with us, tell them early on that it's not a good fit.
Right. I'll just clarify for anyone listening. You mentioned FB, which is what we call it.
Yeah,
FASEB
a little bit of
BioAdvances,
internal
of course.
lingo there.
Yeah, so that was the extra knowledge you gained today. One other thing I'll mention, and just plug the Null Studies special issue that you
That's
and
a
I
great
are co-editing. So maybe talk about the importance of negative findings and how FASEB BioAdvances could be a potential home for those studies.
Yeah, so that has always been a very difficult thing as an investigator myself. When you have this huge study and you put a lot of time and effort into it, and you find something that wasn't what you thought, right? Your hypothesis was wrong, and that's okay, right? But then we always find that it's so difficult to get it published anywhere. There's obviously the positive finding bias that you see in publication, and unfortunately with that, a lot of important work goes hidden to the scientific community, right? We don't know what's already been done and proven to not be right or not be correct. And so it ends up causing a lot of people to replicate ideas that we wouldn't otherwise do. It would already, could already be out there saying, look, this isn't what we think it is, and this is why. So what we like to do is really try to capture those types of studies, null findings, right? It's very hard to get a complete study of a null finding, but there are some really nice ones out there, and we've seen a couple come through that are really important to have in the scientific literature. So if you have a study that didn't turn out the way you think it was, but it's still a rigorous experimental design, you still had all the right controls, and the answer is reliable, and it's just not what you thought it was going to be, FASEB BioAdvances might be a good option for publication of that work.
Great. Well, thank you everyone for joining us today. It's a pleasure to introduce Dr. Sanderson to everyone, and look forward to having many more videos introducing the editorial team and exciting initiatives at the journals.
Great. Thank you, Loren.